to a more sort of specific level. But I think it's really cool to be able to work both locally and at a national level. And so I wanted to hit on that as well. One of them that is, I think, really cool and one of our biggest projects for the last couple of years is this effort working on what we call America's Amazon, the Mobile Tensaw Basin down in South Alabama, which is, as many of you know, one of the most biodiverse places on Earth. Um, and what we've been doing there is working with some private timber companies and Fish and Wildlife Service and in Kazi in a really large effort that's actually going on in a couple places across the country. But we're handling the South Alabama one. It's called WCI, Wildlife Conservation Initiative, and the idea here is that we're getting access to huge chunks of private land on timber company land because there's nowhere else in the country or on the planet, I feel, where biodiversity and timber production comes to such a head as in South Alabama. And so the idea is that if we can document lots and lots of biodiversity there for things like SFI certification, then we can get more landowners on board, more buy-in into certification programs and best management practices and things like that. And so we've been working for a little over 3 years now to try to document basically as much biodiversity as we can in this area. And what's cool about it is that it's a combination of new and old tools done in a way that allows us to collect just tons and tons of data for a lot less money and a lot less resources than was done in the past. And we do this through a combination of new and old tools, really. So things like targeted transects, visual encounter surveys, turtle trapping. So some of those older approaches to documenting biodiversity, but also combining those with newish methods, I'll call them. Things like automated sampling with camera bucket traps, also just regular camera traps, and then throwing in a lot of eDNA. So both methods, qPCR for really rare species and DNA metabarcoding to sort of try to sequence everything in the environment there to try to get a really good handle on what's there for aquatic diversity. And after a couple of years on this project, I think it's really impressive. This is, as far as we can tell and have documented, the largest sampling effort on private lands that's ever occurred in the country, and we believe in the world. But this is across all efforts. We've collected now 2+ years of data, have documented nearly 4,000 individual occurrences, 271 species, and 9 of 31 target species, which are a big long list of SGCN and listed species, and then 7 additional species that we weren't originally targeted that had high S1, S2, G1, or G2 ranks. And this map you can see here sort of shows you the detection across such a broad area using these. So it's really cool. It just kind of demonstrates the level and scale of biodiversity monitoring we're trying to be capable of, as that gets more important, obviously, for everything from just knowing what's on the landscape to species status assessments to the eventual biodiversity credit market. One of the other neat things about this project, and given the scale of it, is that we're putting a lot of different data types together and a lot of different data, metrics and that sort of thing. And so we can compare the effects of sampling methods and start to accumulate data curves and that sort of thing. And we can do this across all species. We can do this for SGCN species or individual species, or that sort of thing. So it's just a really neat large scale project that I thought I would touch on. One of the other interesting projects at a nationwide scale that I think is worth mentioning that also affects North Carolina, and many of you have been partners in some of this, is we've been working on developing genetic databases for turtle species to help put poached individuals back into the wild. And we've been working with law enforcement and a lot of partners for that. And we've had several cases where we've identified where poached animals came from so that they could go back into the wild. And in fact, if you look at this picture here, this is an alligator snapping turtle going back into the wild in Texas from a bust that we were a part of a couple years ago. And we're able to put about 50 alligator snapping turtles back into the wild, and had a successful prosecution on that one because we were able to show that the poachers went across state lines and, of course, broke the Lacey Act and that sort of stuff. And so Heather mentioned the Teddy Roosevelt Genius Prize that we were recently awarded, and it was for this work, creating these genetic databases. And currently we're building those out, or have built those out, for 4 turtle species: alligator snapping turtles, box turtles, bog turtles, and Blanding's turtles. And so it looks like we just haven't been able to progress to C or beyond turtles, anything with a name past C, but we're getting there. We're going to be working on spotted turtles soon, and hopefully a couple others. So it's another big focus of our lab. And one of our other large scale projects is helping DoD monitor and manage imperiled species. So we've been working on a large project looking at building genetic assessments for 9 of DoD's what they call mission sensitive species. These are all amphibians and reptiles, and just looking at sort of critical population monitoring questions, creating lasting genomic resources so that they can manage their biodiversity better across the country. And again, this is right now at about 30 bases across the country, so pretty large scale and neat. And we've been able to work on a lot of really interesting places and species so far. And the last sort of large scale project that I'll hit on, which also includes obviously a important species to North Carolina, and we've partnered with a lot of you on this, is just looking at single species genetic assessments for things like SSAs and species management plans and those sorts of things. This is one that is fresh in our minds because we've been working on this really recently, but the gopher frog, so being reviewed right now for listing by Fish and Wildlife Service. Thanks, Heather. Sorry about that. Warned you it might come. Just didn't think it'd come so quickly. I tried to get it on mute there first, but anyway. So we do a lot of these single species genetic assessments for things like SSAs, and done them on pine snakes and alligator snapping turtles and all sorts of things. This is one that's relatively recent and pretty cool for us. So one of the really neat things about this project to me is that we had over 1,600 samples range wide, so many from partners here in the state. And that allowed us to cover every known population for this species, which is a really special thing. And a couple conclusions or takeaways from this is that there's a major phylogenetic break. So which people have mentioned in the past or found in the past. We've known the peninsular Florida was a lot different. Our data, which is across the genome, shows this as well. And so that's now documented a little bit better. The other thing is that heterozygosity appears to be declining rapidly. So Let's see, this is freezing up on me. So you can see these are heterozygosity levels over the years. One of the other great things about this data set is that we were able to sort of take older samples as well, in those 1600 samples, and look across years, and we have seen a really rapid decline in that heterozygosity, which is really a bad sign for the species. And the other really bad sign for the species is that heterozygosity appears to be lowest in that Florida panhandle. So that has been the area that people have always thought was sort of the stronghold for the species, but turns out heterozygosity wise, they're really struggling. And so this kind of data allows states and federal agencies to create better management plans for the species, um, and move forward. So really excited to be doing that. All right, so let's talk about some North Carolina species. One of our biggest projects and most longest running has been working on the Appalachian cottontail, which is a really neat species that very little was known about, as of a few years ago, and really range wide. There's been very little research. It was split from the New England cottontail only in the 90s. And as far as data on the species, we had very few localities and known in North Carolina, and really very few localities known anywhere except for in West Virginia, where they had a little bit of a better handle on the species. And so, working with you all, working with the commission since 2016 on this species, um, and we've now worked on two different grants. Um, and it's important to get a handle on this. They're Fish and Wildlife Service species of concern across the range, and basically in every state they're in, they're some level of status. And within North Carolina, they're listed as vulnerable, but are also considered a knowledge gap priority species. So this is all the points that we visited or taken samples of during this study. So the purple ones here represent from our first study from 2016 to 2020, and then our more recent version in 23 and 24. You can see we filled in a lot of gaps. So this has been a really massive effort. We've collected over 2,000 scatter tissue samples. We've had over 5,500 trap nights for the species, and done over 200 transects across 75 sites here. Thanks to this recent grant in 23 and 24, a lot of those samples are now on private land. So we're starting to fill in the private land gaps and get a better understanding of overall where this species is and how it's doing. From there. So we've gotten a lot of, obviously a lot of really good and actionable data from this, especially from the first grant so far. We've really learned a lot about their home range size, which appears to be even or pretty small, less than a hectare. And we've learned a lot about sort of the habitat types that they use. High elevation heath balds seems to be a favorite, which wasn't really considered a priority for them before. And we found them at a lot lower elevations than we were expecting. And at those lower elevations, pine and hemlock stands seem to be really important to the species. So starting to get a good handle on their distribution and where they're at, and what they need and what they like. One of the other big sort of tools that we developed for this was a qPCR assay, to identify the species from scat, so to differentiate them from eastern cottontails. And so this really allows us to go to a site and just do scat surveys rather than trapping surveys, which are really difficult for this species. So it's been a lot of good stuff there. On the genetic side, we've been able to do genomic studies from those scat samples, which has been really helpful, and a lot of tissue samples. And it's really interesting, as small as their home ranges are. We can definitely see a lot of genetic structuring here. So places like the Balsam Mountains and Mount Mitchell and Roan are all separated, but not really that strongly. So we see some differentiation, but it's probably relatively recently that they've been isolated. But we are seeing pretty high levels of inbreeding for a mammal species, and so that's worrying, and we're working on sort of figuring that out um, more and more. One of our big pushes over the years and things that are really close to my heart are bog restoration. So this is really working to improve habitat, primarily for bog turtles, but a lot of plant species have been helped by it as well. Um, Putting this talk together, it was really striking to me that we've worked with the commission to restore or do habitat management at over a dozen bogs. So it's really, really neat. Um, really done a lot of habitat work, and I think left a lot of these bogs in much better conditions. So you can see some of those pictures here. One of the cool things we've done is developed a technique for using drones to monitor how we've restored bogs. And so you can see that here. So obviously the top is just simple aerial photography. I always like to use this one because all of the red trees there are poison sumac. So you can see how much fun restoring bogs can be. And I think everybody on that bog restoration ended up with some level of poison sumac, which is not a fun time. One of the other awesome things about using drones for this work is that you can get this elevation data through photogrammetry. And so you can really not just see, but also like sort of quantify how much you've changed the structure of the bog and improved the structure of the bog. Um, we've done two really cool projects with the commission on reducing road mortality. Um, for this was primarily for bog turtles, but for other small animals, obviously, as well. This first one here that you see is the second road passage system in Western North Carolina, the first small wildlife crossing. Sorry, yeah, in Western North Carolina, the first great topped in the state, which is really important for a number of small animals that like to have that light penetrating through before they will go through a tunnel. And this was a really huge project and a lot of multi-agency partners, Fish and Wildlife Service and DOT, and of course the commission. Um, so really, really neat projects to sort of solve some of our problems wildlife wise. And we've also done just fence installs at another site to keep bog turtles off the road. And so just trying to again push conservation forward, find ways to solve the problems that are facing some of these species and get these fences in the ground. Um, and I just included a lot of pictures here of the tunnel crossing because I think it's really neat and that we should be doing it in many many more places across the state. And I always like to just plug this as well because this seemed like a really huge project. I always had it in my head that these sorts of projects would be extremely expensive, but we ended up doing this project for under $70,000. That included the fencing, with the small caveat there that DOT donated their time and expertise in doing the work. But it made me hopeful that we could get these sorts of road crossings for all kinds of taxa done for a lot cheaper than we ever thought possible. One of the other things we work a lot on, and we have some great examples here in North Carolina, is addressing taxonomic uncertainty of species. Across the country, there's probably no better sort of mascot for taxonomic uncertainty than salamanders. One of those places that happens to have a lot of those is about 15 miles from my house, so it's convenient in the Hickory Nut Gorge. And so we've done two projects with Lori Williams looking at taxonomic uncertainty of Blue Ridge Gray Cheek in terms of the range of the species, and the crevice salamander, which is a state listed species, but has long had sort of taxonomic uncertainty whether or not it's actually a species or a subspecies or what it is. So for the gray cheek, we've been able to get a little bit of clarity, and we're continuing to work on these things. And this is, of course, using genomic data. Unfortunately, these things have gigantic genomes, and so it takes a lot of computer power and it takes a lot of time to work through those. But we're starting to get some clarification on exactly where this species is found and where these habitat bound or species boundaries are, and so that's obviously those things are really important for conservation and identifying where they're at and where they're not, and as well as what is a species and what's not. And so this is a great example. Again, the crevice salamander here, the guy on the right was originally described as a species many, many years ago, and then sunk as a species. And so we've been using genomic techniques and working with the museum to sequence mitochondrial DNA, and Brian from the museum, and look at the combination of that genetic data to identify if this is a real species or subspecies or what it is. And this is a little sneak preview of that. It does seem there's some real strong genetic signal there, but final conclusions are sort of still forthcoming. And lastly, I'll hit on another project that I think has been a lot of fun to work on, working on sickle fin red horse, which is a sucker species that's yet undescribed, has been for a couple decades now. And this is a species that's nearly extirpated. It has faced a lot of habitat loss, and has been down to a couple really small populations. And there's been a huge effort from the commission, from you all, from Georgia, and from the Eastern Band of Cherokee, and a lot of folks working to restock it and bring it back. And so our role in this has been to come in and help develop an eDNA assay to go out and sample both where it still could be in the wild, and also to sort of track the success of restocking efforts or lack thereof in many cases. Unfortunately, as anybody who works in fish can tell you, there's a lot of suckers and red horse species out there. And so this was a fun challenge for us to develop it. We ended up having to develop a two-step eDNA assay, one that identified positives and one that split it apart from some of its closest relatives. And so with that approach, we were able to do some really wide-scale eDNA sampling and across Western North Carolina and in North Georgia. And this is what that looks like, which includes basically the entire, or nearly the entire, historic range of the species, and found a few populations that we're still looking into, potentially new populations that were unknown before this, and thankfully was able to pick up on the species or the sites um, where it was unknown, or sorry, where it had been stocked, and so so we was able to show that it worked well in the wild. In other words, and a nice side effect of this as well is that there is some again, a little bit of taxonomic uncertainty with this in terms of was it a real species, and what was its nearest neighbor. So, having all those samples in house, we were able to run DNA. This is um, two studies that sort of contradicted each other in terms of sister species and the validity of it as a species itself. And we were able to run the entire mitochondrial genome for a number of red horse species, including sickle-fin, and also combine that with nuclear data and really sort of refine that the sister species for sickle-fin is actually river red horse, and it is definitely a legitimate species out there on its own. So that's been a really fun project, mainly because the sickle fin red horse group is really active and doing a lot of great conservation work, and so it's an honor to be a part of that. So with that, I'll just say thanks. I wanted to leave a lot of time for questions or conversations. Thanks to all of you in the commission that we work with. I want to give a special shout out to Kate Pipkin, because obviously without her processing payments and that sort of thing, we don't get paid and can't do this work. And obviously we've had a lot of other amazing partners in the commission, and really appreciate that. And of course, all of this work that I presented here is also a reflection of our amazing staff here at Tangled Bank. And we've got a really great team who many of you have interacted and worked with throughout the years. And so wanted to give a shout out to them. And then also just thanks to our many, many partners. Again, the whole point of Tangled Bank was to work and collaborate on conservation projects. And we're really lucky to have so many awesome partners and to make a lot of this work happen. So I want to say thanks to that.
Great. Thanks, JJ. That was a great overview of all the amazing work that you guys are doing. So yeah, guys, go ahead and drop your questions in the chat for JJ. I'll go ahead and start. I was interested in your databases that you're developing for the turtles, and I was wondering if those how wide-ranging those databases are. It looks like maybe they're at a regional level, and if you're collecting all of that data yourself, or do you work with partners to establish those databases?
Yeah, thanks. So the 4 species that we put on there have pretty wide ranges, and for every database, we at least have coverage now across their entire range. So for something like box turtle, that range is from Maine to Arizona, and actually into Mexico. We have that entire coverage. That one we still need to build out. There's still holes in it and that sort of thing. Other things like alligator snapping turtles, we have every drainage where they're found, every major river system. We don't quite have every state yet, only because we haven't gotten it all the way up to Illinois, but that drainage is still covered in other states, so we can still identify that. But yeah, our intent and our point is to have coverage across the entire range of any species we build one out. As far as partners, yeah, this is totally built on partners. And we work with a ton of those. I mentioned a couple, here in terms of Turtle Survival Alliance and CCITT and larger groups that are working on these issues. And then, of course, a lot of groups help provide us with tissue samples and those sorts of things to build those databases.
Oh, thank you. Okay, so I got a question. Is there any post-installation monitoring to see what species are using the crossing structures and how frequently they're being used?
Yeah, great question. I obviously on this platform should have mentioned that the commission is actually handling the post monitoring and I guess a little bit Fish and Wildlife Service as well. But Gabrielle Grader and her technicians have been sort of heading up the post monitoring of the tunnel. I don't think they've gotten any target species yet, but we've seen a lot of things like spotted salamanders and other amphibians using those tunnels so far, or that tunnel.
Great. And then Sarah Schweitzer had a follow-up to that. Could the crossings be a predator trap, i.e., increased predation at crossings?
For sure, and that's a major worry. In fact, one of the things that's come up from that specific tunnel has been sort of the abundance of, especially house cats that we've gotten on those cameras. I don't know really how we get around that. Hopefully things like turtles can avoid adults anyway, can avoid those predators anyway. But that's definitely something we want to keep monitoring and looking into and finding solutions to.
Right. I have another question. If they're speaking about your Appalachian Cottontail project and the transects that you use, you mentioned that a third of those transects were on private lands. So I was wondering what methods you found to be productive in contacting and engaging private landowners for those kinds of studies.
Um, I will be honest, and that's more my field staff doing that. But I know a lot of folks from the commission have had a lot of really great um, contacts that we've taken advantage of, and so it's been a huge help for us. And then the other way we've gotten a lot of access has been through land trusts, and obviously land trusts, hold some land themselves. They have easements, but they also know a lot of folks in the community, and so that's been a really really big help to those efforts and to getting onto private lands.
Okay. And Sarah has a question. Is the technique used on Appalachian Cottontail scat similar to or the same as that used with elk scat?
I don't know what's used on elk scat.
Yeah, I think yeah, so the elk scat where they're doing microsats for population density and abundance, I think that when you're doing the Appalachian Cottontail scat, you were using SNPs, right? So
Correct.
you're using whole pellets.
Yeah, so we're using genomic sequencing for that instead of microsatellites. We're still sort of exploring how to optimize some of that, but yeah, we haven't been using microsatellites for that.
Yeah, so I think they're similar in that you're using the same product, but the methodologies, I think, in the extractions and the downstream marker are different, as well as the applications.